Hi, good morning, everyone. Okay, welcome for S1 and for S6 boys on board with me in this biology lesson. So for those who are not from this class, you are welcome as well. Okay, so once you are with me right now, please get ready your long book. Okay, and I need 4 color pencils as the previous lesson because in this lesson I'm going to teach you on how to draw meiosis, okay, step by step. Okay, next one, for those who just joined, remember to type your name in the chat box as the attendance because I need to take your attendance. Okay, so far I need to check who are here with me. Okay, Jiajun, okay, the first one, very early in the morning, 10:48. Okay, very punctual already with us. Okay, Jiajun, good morning. Jaewon, Shao Qian, Heng Yikang, Qin Rongshen, and why there is the Korean name here, I do not know how to read this, but I just know 안녕하세요. Okay, so please write your name. Don't type Korean language. I do not know how to read. Okay, Chu. Okay, Chu. Manhong, morning. Jen Jun. Junwei, good morning. Ang Yi-sang, good morning. Xin Kai, good morning. Wing Shan, hello. Hong Liang, Jane Hao, Ziyang, Dan Junhao, Ryan, welcome on board. Okay, Chu Jun Sen, Hong Jun Kai, Anthony. Okay, next one followed by after Anthony, Re Rong, Boon Shen, Ng Jun Xian, Chai Yi Kang, Lam Xin Keng, Mei Yong Peng, Calvin Ng, Ang Ziyu, Yap Chi Kit, Hua Kai Wen, Cheng Yao Ming, Yao Ming. Today you have no surprise for you because I'm not going to use your portrait for this lesson, so no worries, Yao Ming. Chan Xijian, Wei Gip, Jia Xuan, Zhao Shen, Ng Shi Hong, Li Ziyang. Okay, I have a friend from Vietnam. Okay, he's joining us. Okay, even though he's not my student, Jeremy. Hello, Jeremy. How are you? Long time no see. Okay, he's from Vietnam. And then Jen Hao, Ong Ning Chow, Henry, and Leong Yuxuan, our ex-student, our senior. Thank you for your support, Leong Yuxuan. He was our ex-student, Form 5 student, class of 2016, Leong Yuxuan from 5S3. Okay, and Wong Jun Chow. Okay, so we start the lesson within half minutes, okay, 30 seconds. Okay, for the rest who just joined, okay, please type your name as attendance because I need to check. And for those who haven't joined, please call your friends, wake up your friends to join our class. And today's lesson, we are going to talk about meiosis. Okay, so even though when I give you the link to join this lesson, there is a thumbnail mentioning that it's a sperm. Okay, right? For this lesson, I am not going to talk about the reproductive system. Very important. Okay, don't misunderstand. Okay, I need to repeat twice. Okay, I'm not teaching reproductive system. I am not teaching reproductive system. Today's lesson is about meiosis, which is the one kind of the cell division. Okay, it's the one kind of cell division. So get ready. The time now is 11:06. We will start our lesson very soon. So please get ready with your long book with you and 4 color pencils. 4 color pencils are for as well as your stationaries, okay, ball pen and pencil. Okay, so get ready. So before starting the lesson, please make sure you have taken your breakfast, okay, because our class will end around 12:30 to 1, okay, or else you bring along your food, sit in front of your laptop or sit in front of your mobile phone. Okay, so welcome everyone. Okay, good morning. Chun K and Sean Chong, ex-student here. Sean, there are so many students with surname Chong. I'm not sure who is Sean Chong. It's okay. So we will start the lesson right now without further ado. Okay, so the attendance, I see 35 so far. 2 classes add together should be 468. Okay, so it's no worries for those who are unable to join because I believe maybe they are unable to connect to internet, okay, or they share devices with their family members, and no worries, because for today's lesson you can replay anytime. Okay, so before starting the lesson, okay, I would like to let you to do simple calculations for mathematics. Okay, are you ready? Okay, so you know that for the chromosomal number in a normal human cell, okay, which is 46, right? 46 chromosome in a human cell. Now, let's say last lesson we talked about mitosis. Okay, mitosis is a cell division from one cell to two cells. Okay, let's say if one cell is divided to two cells, initially the parent cell has 46 chromosomes. Okay, so once the parent cell is divided into two daughter cells, okay, let's see. When 46 divided by 2, so the answer should be 23, right? But in mitosis, the chromosomal number in the daughter cells are same as the parent cell. So which means, okay, if let's say the parent cell has 46 chromosomes, which means in the daughter cells, they also have 46 chromosomes. But through this calculation, if one cell is divided by 2, 46 chromosomes is divided by 2a. The answer should be 23. 23 is not equal to 46. So this is the topic to be discussed. Okay, so in mitosis, okay, now for those who just joined, I'm just introduction regarding to the previous lesson, mitosis, no worries. Okay, I will enter meiosis very soon. Okay, so therefore, initially, the parent cell has 46 chromosomes, but do not forget, before mitosis, a cell is at its interphase. So in interphase, there is an S phase, S phase. So in S phase, DNA replication occurs. Okay, DNA replication occurs, and now there are 46 times 2, which is 92. Okay, it doesn't mean the cell has 92 chromosomes. No, no, no. The cell remains as 46 chromosomes, but there are 92 copies of DNA. Okay, it's double. So this is the reason why DNA replication takes place. And now, okay, during mitosis, the 92 so-called, okay, genetic information, okay, or the DNA copies is divided by 2. Okay, so remember, in the parent cell, there still consists of 46 chromosomes. Okay, it's not 92 chromosomes. This is just the genetic material because of the DNA replication. And now, in each daughter cells consists of 46 chromosomes. Okay, so now I go to another calculation mathematics question. For those who joined the lesson, so remember to type your name in the chat box as your attendance. Okay, let's check who are the new one. Okay, Yik Sun, good morning, and Liu Chila, good morning. Okay, so for today's lesson, we'll talk about another type of cell division, which is known as the meiosis. Okay, so for meiosis, why meiosis takes place? Let's see. This is related to the sexual reproduction. Yes, even though today's lesson is not about sexual reproduction, but I will talk a bit, okay, related to sexual reproduction. So during the sexual reproduction, so the male gamete fuses with the female gamete, okay, to form a zygote, and then the zygote grows, okay, to form embryo, fetus, okay, after a mother gives birth to the baby, from the baby to a child, from a child to a teenager, and then to an adult. Okay, let's say, if our sperm contains 46 chromosomes, okay, in the cell, and let's say the ovum consists of 46 chromosomes. Okay, so this is a very simple calculation. 46 plus 46, which is equal to 92. So if a cell with 92 chromosomes, this is not a normal human. Okay, now if this is due 92 chromosome, fuses with the ovum of your wife in the future now become, wow, 184 chromosomes in the cell. So this is no longer a normal human, it's a superhuman, unless we are a Superman, just like me wearing the Superman cap today. Okay, so this is impossible, right? And therefore, this is the importance of today's topic about meiosis. So in meiosis, the 46 chromosomes, okay, initially, 46 chromosome in the human cell is divided by 2 to become 23 chromosomes. And in the female gamete or the female reproductive system, the 46 chromosomes is divided by 2, which means the number of chromosomes must be reduced by half, okay, 减一半 to become 23. And now, when the sperm fuses with the ovum, and now the number of chromosomes of the species is maintained as 46. This is only for human. Is it clear? Okay, so we'll start the lesson right now regarding to this process, which is known as 46 divided by 2. This is known as the meiosis, which is one kind of the cell division, different from mitosis. Okay, so good morning to Lam Tian Pang. Okay, and who is the medic here? Okay, so I will share the screen while waiting for me to share the screen. Okay, so please get ready with your longbook. Okay, yesterday I just shared with you in your group, okay, in our group, the format to draw the tables on your longbook. Okay, so please according to the framing format. Sorry. Okay, so for those who just joined the lesson, remember to type your name in the chat box as attendance. And do not forget to have your breakfast before the lesson, or while having the lesson also you can eat. And make sure there is a cup of water beside you because you need to keep yourself to be hydrated from time to time. Okay. So for today's lesson, same as the previous lesson, we need to learn on how to draw. Okay, I think you can see my face together with the slide. Oh, cool. Okay, so the sperm is asking me, sir, how am I formed? So a sperm or reproductive cells, they are formed through a cell division known as meiosis. Okay, so today's lesson we are talking about meiosis. Okay, let's see this diagram. What's the difference between mitosis and the meiosis? Okay, sorry, I need to check the chat box at the same time. Okay, because I'm using the handphone to check your attendance. Okay, so just let's see. Okay, so what is the difference between mitosis and meiosis? As we know, meiosis is a cell division to produce the gametes or the the reproductive cell, okay, the sperm and the ovum. Okay, let's see. If the man, okay, we have 46 chromosomes in our somatic cells. So which means our cell is known as a diploid cells, 2N. 2N here means what? Which means there are two sets. One set of chromosome is given by our father, and one set of chromosome is given by our mother. So in total, there are two sets. Same goes to the female. There are two sets of chromosomes, 46. From these 46, 23 chromosomes are contributed by the father and 23 are contributed by the mother. So altogether it's 2N. Okay, so but as I told you, in order to maintain the number of chromosomes in the same species, like in human, we must maintain the number of chromosomes as 46. So in meiosis, the function of meiosis is to reduce the number of chromosomes by half. Okay, so through meiosis, okay, the sperm cell now contains 23 chromosomes, okay, which is the haploid number, haploid. For 2N is diploid. Previously, I have told you the meaning, okay, the difference between haploid and diploid. Haploid means one set, diploid means two set. So same goes to the female. Okay, same goes to female. It is the cell, okay, all the somatic cells, they are the diploid cells. So now, the diploid cell undergoes meiosis to form a haploid cell, which is the ovum. So once the sperm, okay, fertilize the ovum, okay, so it will form. Okay, now before fertilization, this process must be very important. Okay, this is the copulation. If no copulation, how to form the zygote, but we still have a test tube baby. There are many technologies right now. No need to undergo this, carry out this copulation. We still can form a zygote. Okay, we can use other methods. Okay, we can use technologies. Okay, now let's say once the sperm fuses with the zygote, okay, and this process is known as fertilization, right? So to form a zygote, okay, which is 2N. Okay, N plus N is 2N. So now the zygote grows, okay, divides into 2 cells, stage 4 cells, stage 8 cells, and then to an embryo. So this is known as mitosis. Keep dividing, okay, and to form a baby. Okay, after giving birth, so the baby grows to become an adult. So this involves mitosis. So for meiosis, this term is only for the production or the formation of reproductive cells. For the other cells, the somatic cells, okay, somatic cells are all body cells except the reproductive cells. So for somatic cells, they carried out mitosis only. So we can see from this diagram, for mitosis, the number of chromosomes in the daughter cells is same as the parent cell, from 2N to 2N. But for meiosis, the number of chromosomes in the daughter cells is half of the parent cell, from 2N to N. Is that clear? Okay, so this is a clear image regarding to the difference between the mitosis and the meiosis. Okay, so for today's lesson, I want you to prepare a cup of water, but not a full cup of, or not a full glass of water. So just half glass will do. Same goes to today's lesson. When you're going to learn meiosis, I want you to half fill your glass. So this glass of water, which means your prior knowledge, you need to know what are homologous chromosomes, as I introduced in the previous lesson. So the homologous chromosomes are the pair of chromosomes, and they are similar in terms of their structural characteristics. They have the same height and the same positions of the centromere. Okay, so another one, which is sister chromatid. In each chromosome, there are 2 sister chromatids. Okay, you just refer to this diagram. Okay, the purple color and the green color. So for the purple color, they have left side and right side. So the left and right, there are sister chromatids, and then they have the same copy of DNA, left and right, same DNA. So both of them are joined at the centromere. So, and then the prior knowledge you need to know is the centroles, and the function of centroles is for the formation of spindle fiber. So what is the function of spindle fiber? Is to separate the sister chromatids. In the last lesson, the function of spindle fiber is to separate the sister chromatid. Okay, when you see spindle fiber, SP, you relate to separate. Its function is to separate. Okay, and then we have PNAT, okay, and the cytogenesis. And so this is the prior knowledge, which is the half-filled glass, okay, with water, and the empty part, which is your new knowledge about meiosis. So you need to remove water from your glass first. Let it half fill, so you only can have some input for today's lesson. Okay, so we will start our lesson right now officially. Okay, let's see this question. Okay, if let's say your parents ask you, boy, where are your sperms? What should you answer? What should you answer? In the toilet or my tissue paper? Okay, so the proper way, after studying biology, you should tell your friends, okay, or your parents, oh, my sperms, they are in my test right now. Okay, so for testes, the spelling of testes is T-E-S-T-E-S. Testis also can be accepted unless you have one ball. If one ball, this is testis. If a pair, must be testes. No three testes. Okay, everyone two testes. This is normal. Okay, so one testis, two testes. So the sperms are formed in your testes. So as I told you, for the formation of sperms, the parent cell, okay, contains a diploid number. Okay, like human, we have 46 chromosomes, okay, as a diploid cell. Now, okay, so the diploid cell here are known as the primary spermatocytes. Okay, the diploid cell here, primary spermatocytes. So in our testes contains many primary spermatocytes, and these primary spermatocytes, okay, they undergo meiosis to form sperm, which are haploid cell. Okay, so remember, okay, don't just measure cells of testes. There are various kinds of cell in the testis, not just contains the sperm, but the diploid cell or the parent cells, they are known as the primary spermatocyte. Is that clear? Okay, so now, okay, sorry. So let's say, if for a female, a girl, where are your ova? Okay, what is ova and ovum, the difference? Just a grammatical difference. Okay, so for ovum, this refers to singular, one ovum. But for ova, this is many, plural, many ova. Okay, so the site of production of ovum is actually ovaries. So for a normal female, they have two ovaries at the left and at the right, okay, both sides. So the ovaries, there are various kind of this, they contain these cells known as a primary oocyte. Okay, so you can see there is a similarity between the diploid cells in the ovaries and the diploid cells found in the testes. Okay, the term, the similarity is they start from primary, right? So primary for sperm, primary spermatocyte, and this one is primary oocyte. Okay, so now the primary oocyte, which are the diploid numbers, okay, diploid cells, and they will undergo meiosis to form ovum, which has the haploid number. Okay, so now why we need to carry out the meiosis? Okay, so for last lesson, I believe all of you would answer the same question, right? Which is for the formation of gametes. Okay, so through gametogenesis. Now, that is a question for you, and you need to think, hey, sir, why production of gametes is not known as meiosis, but now there is another term, gametogenesis. That is a difference. Okay, so for the whole process, okay, to produce gametes is known as the gametogenesis. Okay, on your side, please mention once again gametogenesis for 3 times. Gametogenesis, gametogenesis. Okay, so please be excited until your parents know you're studying biology right now. Wow, very excited. So now, but for meiosis, what is that? Meiosis is just a cell division. From one cell is divided into 4 cells. Okay, but gametogenesis is the whole process. And we need to make sure the diploid chromosome number, okay, of a species is maintained. Just like for human, we have 46 chromosomes. If let's say the sperm has 46 chromosomes and the ovum has 46 chromosomes, altogether is 92. Impossible to be 46. No, we need to make sure same, unless you want to be superhuman like Superman, the cap I'm wearing right now. Okay, so the next one is to produce genetic variation in the offspring of the same species. Okay, so we just compare. Okay, use your siblings to compare. So your brothers, you, okay, and your sister's parents are the same. Okay, from the same mother. Okay, you have the same mother, same father, but you have different physical appearance or even different blood group. So these are all known as the genetic variation. So because of meiosis, this can produce the variations among the offspring of the same species. Okay, so now we will enter meiosis right now. And please, boys, please get ready with your long book and 4 color pencils with different colors. okay, because we need to follow the previous lesson. Okay, I'm going to teach you on how to draw meiosis. Okay, so if you are ready, everything with you, okay, your long book, okay, your stationaries with you, please type ready in the chat box. Let me check. Okay, if you are ready, please type ready. Okay, so if, or you can just shout, hey sir, I'm ready right now, I'm ready right now. Okay, until your parents know you're studying biology, you're very excited. Oh, my son really likes biology. If you're very shy, introvert, okay, you don't dare to, okay, shout, you just type here, I'm ready, sir. Okay, and I'm ready to teach. Okay, fine. Well, now we'll go to the next part, which is meiosis, since most of you are ready for the lesson. Okay, if you are not going to type, just shout it at home. Okay, so meiosis, unlike mitosis, they just consist of mitosis, just mitosis. But for meiosis, there are two main phases, the meiosis I and the meiosis II, 1 and 2. So for meiosis I, it involves a separation of homologous chromosomes. Okay, in the previous lesson, mitosis, we never mentioned about homologous chromosomes in mitosis, just separation of sister chromatids will do. But for meiosis, okay, we need to mention, okay, we have to mention the homologous chromosome. Okay, as I told you previously, in meiosis, the number of chromosomes is reduced by half, okay, which means from 46 chromosomes in the somatic cell to 23 chromosomes in the gametes. Okay, so it involves separation of homologous chromosome, which means in meiosis once the diploid cell now become the haploid cell from 2N to N. So this is the significance of meiosis I. So whereas for meiosis II, it's just a separation of sister chromatids. Okay, so now we'll go to meiosis I. So for meiosis I, same as the mitosis, we know there are P, M, A, T, prophase, meta, ana, and telophase. But in order to differentiate meiosis I from mitosis, name is same, but because we need to differentiate meiosis I and mitosis, and therefore we need to write one just behind the term prophase I, metaphase I, and so on. And same goes to meiosis II. Okay, got prophase II, metaphase II, and so on. Okay, so now at the same time, if you have textbook with you, you can take out your textbook right now, because for my meiosis, okay, the diagram, actually I'm using the diagram from the textbook. Okay, so before a cell in the reproductive system, okay, I take man as example. So before a primary spermatocyte, okay, undergoes meiosis I, so the cell is at the interface, as I've told you previously. So in this interface, what happened in this what happened in this interface? Okay, so the cell grows, okay, and in G1 phase, G2 also the cell is growing, and now the DNA replicates. Okay, remember, same as mitosis, replication of DNA occurs. Okay, so now the cell enters meiosis I. So in prophase I, okay, let's see what happened. Okay, so get ready with your 4 different colors. Okay, in the previous lesson, I've told you I'm using the blue color, the blue pen. But for your side, you can use whatever color you want, but do not use, for drawing, do not use a ball pen to draw. Okay, I allowed you for this time to use the color pencil. Okay, I use the blue pen to represent the chromosomal behavior. Okay, and next I'm using the red pen. Red pen represents what happened to the nuclear membrane, nuclear membrane and the nucleus. Okay, and then I have the spindle fiber. Okay, purple color represents what happened to the spindle fiber, and this orange color represents the centrioles. Okay, so first in prophase I, everything for this slide, everything is related to the chromosomal behavior. Okay, so I'll start now and I will slow down a bit. Okay, so for in prophase I, the first step, okay, it is similar to prophase of mitosis. Okay, the chromatins condense, Okay, to form chromosomes, and they become shorter, thicker, and they are visible under like a microscope. Okay, it's exactly the same. But now the difference, okay, when we refer to this diagram, okay, so you can see that is the sister chromatids, okay, they overlap each other, which means this is the event occurs in prophase I. Okay, so the first one, this is special case for special. For the first one is normal. You just follow Prophase I. Okay, so you just remember, you just memorize the one fact, one sentence, but for two processes. Okay, very convenient for you. But for this one, you need to spend some effort, some time for this. Okay, let's see. Synapsis of homologous chromosome occurs. Okay, synapsis here means the pairing. Okay, in Prophase I, the homologous chromosome, they must be paired together. So, and this pairing process is known as a synapsis. So once the homologous chromosome, okay, they are paired side to side, okay, left and right. So they form a bivalent. Bivalent, which means two homologous chromosomes together, left and right. Process is known as a synapsis. Okay, so let's see. By referring to this diagram, okay, the sister chromatids, they overlap right now. But we are not going to say they overlap. Instead of that, we use crossover. Okay, so for this 21st century, this crossover we always must implement in our daily life, like biology. We can cross over biology and mathematics and maths. Okay, like if you are in one society cross over with another society, because we need to exchange, right? Exchange our idea, exchange the knowledge. Same, the purpose of crossing over is to exchange. Okay, let's see the term genetic materials. So this crossing over, where does it occur? Between the non-sister chromatids. So non-sister chromatids, I will show you what is the meaning of non-sister chromatid. Okay, so now give yourself 10 seconds crossing over. Where does it occur? Non-sister chromatid. Why does it occur? To exchange genetic information. Okay, so let's see. So both of these are the sister chromatids. Are the homologous chromosome. Homologous chromosome. One is the maternal origin, another which is the paternal. Okay, so in the previous lesson, okay, we have studied about, okay, in each chromosome, there are two sister chromatids, left and right, okay, and joined at the centromere. But now, this crossing over occurs between non-sister chromatids. Non-sister chromatids means different leg, your leg and my leg. So your leg kick my leg. This is crossing over. So not the same sister chromatid, but different. But this non-sister chromatid must be the homologous chromosomes. Okay, and now they cross over. Okay, and the point where they cross over, okay, you can see the point, the point where they cross over. This is known as a chiasma. Okay, so now the genetic information from one sister chromatid, okay, is exchanged with another sister chromatid, but they must be non-sister chromatid from different chromosomes. Okay, so now they exchange. So this is how they look like, okay, after crossing over. So the blue color part goes to, okay, the one part of the blue color one, okay, goes to the white color, and the white color one part goes to the blue color. Okay, so for meiosis, okay, we need to use, okay, different colors to differentiate, okay, the homologous chromosome. Okay, just like this, there is a pair of homologous chromosome. So let's say if I draw this prophase, okay, with both homologous chromosomes same color, okay, even though there is crossing over, but we can't see which part, okay, whereby the genetic information has been exchanged, and therefore we need to use, okay, one is darkened, another one is the non-darkened one. Okay, is that clear? So the next part would be the nucleolus, okay, the centroles, as well as the spindle fiber. Everything remains same as in mitosis, as long as you have the prior knowledge of mitosis. So once you come to prophase meiosis, you have no problem. Okay, So for nucleolus disappear, nuclear membrane disintegrate, which means the nuclear membrane is broken into smaller pieces. Okay, and the centrals that start to move to the opposite pole. So for the opposite pole, okay, I repeat, okay, opposite pole means left and right. They are opposite poles. Okay, top and bottom, they are opposite poles. And spindle fibers start to form. Everything same as in prophase of mitosis. Okay, so now I will proceed to metaphase. Okay, M means middle. So all the chromosomes that come to the middle of the cell, and this middle of the cell known as the equatorial plane or metaphase plate. Up to you, both also can be accepted, same meaning. Okay, so by referring to this metaphase one, the diagram, okay, we know that all the chromosomes, they come to the middle of the cell, but the difference is, okay, we can see the difference, they are not, all the chromosomes, they are not in a single row, but instead of that, they are in pairs. Okay, let's see. Now, the centrals that reach the opposite pole of the cell, okay, left and right, up and down. And the spindle fibers, they are fully formed right now. Okay, we just follow the metaphase. Okay, so the homologous chromosomes, okay, the difference is the homologous chromosome, each pair, the pair of chromosomes are arranged side by side, left, right, left, right, left, right. Okay, at the equatorial plane, no longer in a single row, but they are arranged in pairs, so homologous chromosomes. And now, one chromosome, okay, you just refer to any, there are 4 chromosomes, right? Okay, you just refer to one chromosome, you do. So one chromosome of each pair is attached to the spindle fiber from one pole. Okay, so which means that the chromosome, the sister chromatids are still joined together at the centromere. There is no separation. Okay, so this is about metaphase. The difference is homologous chromosomes arranged side by side. Okay, next I will proceed to anaphase. So as you know, for meiosis I, its function, its role is for separation of homologous chromosomes. Okay, which means a diploid cell now becomes a haploid cell from 2N to N. So for anaphase, it involves the separation of homologous chromosomes. Okay, by the shortening and the contracting of spindle fiber. So once spindle fiber shorten and contract, so the homologous chromosomes are pulled to the opposite pole. So in this case, there is no separation of sister chromatids. Okay, by referring to the diagram, in each chromosome, okay, in each chromosome, the sister chromatids are still attached, okay, and joined at the centromere. There is no separation. That just involve the separation of homologous chromosomes. Okay, so this is about anaphase I. It involves the separation of homologous chromosomes to the opposite pole. Okay, so next one for telophase. For telophase, okay, now the chromosomes reach the opposite pole. Okay, the nucleolus reappear. Okay, nuclear membrane are reformed, same, and the spindle fiber disappear. Okay, so for this telophase, the chromosomes, okay, we are in the previous lesson about mitosis. In telophase, the chromosomes, they decondense or they uncoil to become chromatin. But in telophase I, the chromosomes do not decondense, because after telophase I, it will enter another phase which is known as the prophase II. So in this telophase I, the chromosomes do not decondense or do not uncoil to form chromatin. Okay, so just once after telophase I, okay, the cell will proceed to cytokinesis, which involves the separation of the cytoplasm. And then immediately we'll proceed to meiosis II. Okay, so most of the cells, they are not at the stage of interphase between telophase I and meiosis II. Okay, which means from there, after cytokinesis, the two daughter cells, they are formed, and they will proceed to meiosis II, okay, without undergoing or carrying out the interphase. But some organisms, they do have interphase before entering meiosis II, but the time is very short. But most of the cells, they are not in the state of interphase before meiosis II. Okay, so before moving to meiosis II, now I do not want you to keep listening to me. Now it's a hands-on activity. Okay, I want you to take out your longbook and we need to learn on how to draw meiosis I. Okay, please get ready. Waiting for the others. Okay, so please follow the framing format. Okay, so we assume that the cell we are going to draw right now, the diploid number 2n, which is equal to 4. 2n, which is equal to 4. If you follow human cell, 46 chromosome, you draw R until next day also cannot finish. Okay, so we made it simple, 2n, which is equal to 4. So we start from Prophase I. Okay, so we are ready. 4 different colors of color pencils. Okay, these 4 colors represents what? Okay, I repeat once again. For your chromosomal behavior, nuclear membrane, nucleolus. Okay, and then for the spindle fiber as well as a central. For the plasma membrane, you just use pencil will do. Okay, so now let's start. So we start from the Prophase I. Okay, the plasma membrane. Okay, and then we know that in this phase, now, for nucleolus, nuclear membrane, it only involves the first prophase and the telophase. In between the metaphase and the anaphase, we do not draw the nucleolus. Okay, so you just need to know 包头包尾 for the nucleolus and the nuclear membrane. So now the nuclear membrane is integrated, which means it is broken into smaller pieces. And then nucleolus disappear. Okay, I'm now using the red color one to represent the nucleolus and the nuclear membrane. Okay, and then for the centrals, okay, because in the S phase, the centrosomes duplicate S phase, and spindle fiber begins to form, just like drawing the whiskers of a cat. I'm using purple color, but you use your own color. Okay, so for chromosomes, okay, now the last step I want you to draw, because for the other parts, we just followed prophase of mitosis will do. But the difference is in Prophase I, the homologous chromosomes that are paired to each other by a process known as a synapsis. So once they are paired to each other, they form a bivalent, which means 2 homologous chromosomes stand side by side. Okay, so when I draw the homologous chromosome, one which is not the darkened one, okay, another which is the darkened one. Make sure, okay, so to differentiate which one is the maternal origin and which one is the paternal origin. Okay, so now overlap, we are not going to say overlap, it's a crossing over. So now crossing over takes place. Okay, we just cross, okay, you see this part, crossing over takes place. So we assume that this cell 2n, which is equal to 4, 2n, this is the diploid cell. Okay, which means there are 4 chromosomes. So another chromosomes, okay, I draw a shorter one, another pair. Sorry. Okay, so there is a crossing over between the non-sister chromatids, different legs. Okay, and a function, the role of of crossing over is to exchange genetic information. So this will produce genetic variation in the offspring. So this explains why, okay, your characteristics, okay, even no matter this physical characteristics or the chemical characteristics is different from your siblings because of the crossing over takes place in Prophase I to exchange genetic information. So this is about Prophase I. Okay, now I will proceed to Metaphase I. So for Metaphase I, okay, so after exchanging, I draw at this side, okay, see it. So this is one homologous chromosomes. Okay, I just take this as an example. So on the left is the undarkened one, and this is the darkened one. So when they cross over, so this tip is changed. Tip has been changed. Okay, you can see. For this tip is not a darkened one because the genetic information is exchanged from this non-sister chromatid. Okay. On the other hand, this tip is darkened. So this is the result of crossing over. So later, when we enter metaphase, make sure when you draw the homologous chromosome, they are in this form. Okay, so now we go to metaphase. Okay, now get ready. So in metaphase, there is no more nuclear membrane because it has been broken into smaller pieces, and now the centroles reach the opposite pole. Centroles reach the opposite pole. Okay, and red. So use different color to represent the centroles. For me, I'm using the orange color, and for your side, you can use any color you do. Up to you. As long as from the drawing you can see 4 different parts. Now the spindle fiber are fully formed. So now, since the opposite poles, they are at the up and down, top and bottom. So when the homologous chromosomes, they are arranged side by side at the metaphase plate, right? So the metaphase plate would be this one. This is the metaphase plate. So you no need to draw the metaphase plate. This is just an imaginary line. Okay, it does not exist. Okay, later I will erase this part. So the homologous chromosome side by side. Okay, here I have one. Okay, this is for the longer the longer one, and this is for the shorter one. Okay, now I erase this part. Okay, can you see this? Okay, since you know that for homologous chromosomes, when they arrange side by side at the metaphase plate or the equatorial plane, they are arranged randomly. Okay, which means now this is one pair of homologous chromosome. Okay, so let's say I say this A and B, A and B. A can be at this upper part, B can be lower part. Or in opposite way, A can be down, B can be up. It's okay because they're arranged randomly. Because of this random arrangement of homologous chromosomes, okay, in the metaphase state, this can produce the genetic variation. Okay, so one is a darker, another one is not darker. But remember, okay, can you see this? For this part, this sister chromatid, this tip has been exchanged with this sister chromatid. Same goes to this one. Okay, so here, this part has been exchanged, the genetic materials has been exchanged. So for this tip is the undarkened one, and this tip is the darkened one. Okay, so clear? This is about metaphase 1, whereby the homologous chromosomes, they are arranged side by side at the equatorial plane or the metaphase plane. Okay, so now I will proceed to the next one, which is the anaphase. Okay, so for those who have completed before, while waiting for the others, okay, or while waiting for me to proceed to anaphase 1, so you can draw the outline of the plasma membrane for the anaphase 1. So as we know, anaphase it involves the anaphase 1. It involves the separation of homologous chromosomes to the opposite pole. So when you draw the outline of the plasma membrane, it must be an elongated one. Okay. Ready? So now I'll proceed to another part, which is anaphase 1. So for anaphase 1, the cell is elongated. Okay. And now centrals, don't forget, at the opposite pole. Okay, so now next one would be the separation of homologous chromosomes. So for separation of homologous chromosomes, okay, let's see. The homologous chromosomes are separated. Now, initially, for these chromosomes, when they are arranged at the metaphase state, okay, is this way right? For this homologous chromosome, this is the shaded one. Okay, so now when both of them separated, the homologous chromosome, the chromosome is led by the centromere, which means the centromere is facing towards the central, Okay, initially is the flattened one, and now they become a V shape, and pull to the opposite pole. And same goes to this one. Okay, so don't forget this part. Okay, so remember it's a V shape, and the chromosome is led by the centromere, while it is attracted to the centrals. So if I'm too fast, please type a comment here and slow down. If you cannot follow, it's okay. Never mind, you can keep replaying. Okay, the next one, another homologous chromosome. This one is a shorter one. Okay, so make sure this part, because the genetic information has been exchanged. So make sure one tip is darkened for this, and this part is not darkened. And don't forget the spindle fiber because of the contraction and the shortening of spindle fiber. So this causes the separation of homologous chromosomes to the opposite pole. Okay, that clear? Okay, next one I will proceed to telophase I. So in telophase I, the chromosomes, okay, after separation of homologous chromosome, we no longer mention homologous chromosome because they are already separated. So the chromosomes reach the opposite pole. So initially the cell has 4 chromosomes, and now the number of chromosomes is reduced by half into 2 chromosomes in each pole. Okay, so now we'll proceed to another one, which is telophase I. So for telophase, when we draw the plasma membrane, make sure at the middle of the cell, which means at the equatorial plane, the plasma membrane is a bit curving inwards. Just like drawing the groundnuts. Okay, so now in telophase I, the nuclear membrane reforms and the nucleus reappears, and the spindle fiber and the central as well, don't forget. So this is an animal cell, okay boys. For plant cell, they do not have centrals, so you must make sure that okay, this cell is what type of cell, whether this is a plant cell or an animal cell. Okay, now the chromosomes that reach the opposite pole, so one long, one short, one long, one short. Okay, so for previous one, okay, for anaphase one, because of this darkened one is go up, the darkened one is going down, right? The darkened one is going down, so this is darkened one. And don't forget, this tip is not the darkened part because genetic information has been exchanged. Same goes to this. Okay, and how about this one? So this is telophase I. Okay, so the time now is 12:01, and I still have a small part, a little part, which is regarding to meiosis II. Okay, so before proceeding to meiosis II, I will give you a 3 minutes break starting from right now. Okay, if you wish to go to toilet, you may go now. Okay, and if any part you have missed, you can replay. Okay, so 3 minutes break start from right now, and I will start meiosis II after 3 minutes. Okay, so see you later. Okay, hi everyone, welcome back. Okay, so for the next part, I'll talk about the Meiosis II. So I will not spend too much time from you, and hopefully we will start end the lesson at 12:30. Okay. Okay, so while waiting for the others to join, okay, come on, 5 minutes is over, or 3 minutes, 3 minute break time is over. Please come back to the lesson. Come on, everyone. Where are you all? Or you're going to toilet to check your sperm? Okay, everyone, come back right now, come back right now. Okay, next part we'll proceed to the Meiosis II. Okay, I'm not going to wait for the others because as I said, 3 minutes break, then 3 minutes break. Okay, so get ready. Okay, I'm going to share screen. Okay, so last topic we stopped at the telophase one, okay, whereby a parent cell, okay, is separated or is divided into two cells from one cell to two cells. And then the number of chromosomes initially is 4, and now is reduced by half into 2 chromosomes in each pole. So the significance of meiosis I is for the separation of homologous chromosome and the reduction of number of chromosomes by half. Okay, so that is a question for you. In each pole, how many chromosomes are there? Okay, just now we have drew the diagram whereby there are only 2 chromosomes, okay, in each daughter cells, and now the cell is known as the haploid cell. Okay, boys, if there are two set means the diploid cell. If now become one set of chromosome, this is the haploid cell. Okay, so after telophase I, a cell will undergo cytogenesis, whereby the cytoplasm is separated and now one cell is divided into 2 daughter cells. But in meiosis, it contains 2 phases, the meiosis I and the meiosis II. So which means a parent cell divides for 2 times. The first time is in meiosis I, and the second time is during the meiosis II. Okay, so now in meiosis II, the stages are almost the same: prophase, PMAT, prophase, metaphase, anaphase, telophase. But we need to add 2 behind the name in order for us to differentiate from mitosis and differentiate it from meiosis I. Okay, so which cells will carry out meiosis II? Okay, previously there are 2 daughter cells are produced, okay, at the end of meiosis I, right? So these 2 daughter cells will proceed to meiosis II, okay? And in meiosis II, let's see, same, we have prophase, metaphase, anaphase, and telophase 2. Okay, in prophase II, what happened? Okay, boys, if you have the prior knowledge regarding to the mitosis, that's why I've told you from this lesson, you must be half filled, your glass must be half filled with water. The half filled part of water, which is your prior knowledge, and the empty portion, which means I need to pour water into it, which means the new input for you. So your prior knowledge is very important. Okay, so for Prophase II, it is not Prophase II, for Meiosis II. So from the beginning until the end, so it's 90%, I can say 90% is same as mitosis, but the only difference is the drawing. Okay, now you see. Since in telophase I, the chromosomes that do not decondense or they do not uncoil, they remain as the thicker or the shorter form. So which means in Prophase II, we don't need to mention about what happened to the chromosomes. Okay, so the nucleolus disappear, nuclear membrane disintegrate, okay, or broken into smaller pieces. Centrals move to the opposite pole. Okay, and the spindle fiber start to form, you see. All of this, same as prophase of the mitosis, same thing, same sentence, okay? So in this case, now the chromosomes now is 2 chromosomes in the cell, which means this is a haploid cell. And each chromosomes, in each chromosome, the sister chromatids are still attached to each other or still joined at the centromere. Okay, there is no separation of sister chromatid in prophase II. Okay, so we don't need to mention about the chromatin condensed to become chromosomes, no need. Because in Telophase I, there is no decondensation. Okay, now we enter metaphase II. So N metaphase means all of them will go to the middle of the cell. N means middle or the cell equator or the equatorial plane, or simply known as metaphase plate. So now, after meiosis I, we no need to mention about homologous chromosomes, okay, because they have been separated. So in metaphase II, okay, centromes reach the opposite pole, right? And the spindle fiber are fully formed, same, exactly the same. And the difference is the chromosomes, no need to mention homologous chromosomes because they have been separated. Okay, so chromosomes are arranged randomly at the equatorial plane. So this is about metaphase II. Okay, so one more thing I need to mention, I need to correct, okay, our textbook. Okay, textbook, everything, the diagram is very pretty, okay, the information is good, but the problem is, okay, in meiosis, the separation, okay, in meiosis I and meiosis II, the movement of central is in different direction. Okay, according to our previous part, which means before the break, before the break would be the meiosis I. For meiosis I, when I taught you on how to draw the cells, the stages, the centrals that move to the opposite pole, which is up and down, right, up and down. But in meiosis II, okay, the movement of centrals would be in different directions, go to left and right, left and right. So this is little imperfections in your textbook, but it's okay. Okay, as long as you know in a cell. Okay, let's say later I will draw this on the board for you, and you know why the movement of centrals is in different directions. Okay, which means in meiosis I and meiosis II. Okay, so now we proceed to Anaphase II. Anaphase means separate, pull, they go far apart from each other. A means apart, 离开. Okay, in Anaphase, sorry, this is a typing error. Should be Anaphase II. Sorry. Okay, so in Anaphase II, what happened is a separation of sister chromatids. Do not mention homologous chromosomes again. Okay, so the spindle fibers shorten, contract, and the shortening and the contract of spindle fibers causes sister chromatids to be separated. Okay, now the centromere divides. Okay, remember when we mentioned about the separation of sister chromatid, first of all, we must describe what happened to the centromere, because the function of centromere is to join the sister chromatids together. Okay, so now before they separate, the centromere now divides, and the sister chromatids of each chromosome, they are pulled to the opposite pole. Okay, there is another mistake here. Sorry, PUU. Okay, pulled to the opposite pole. Okay, so there's nothing happened regarding to the nucleolus because nucleolus disappeared, and the nuclear membrane is already broken down. So you don't need to mention what happened to it. And also same goes to the central, because once the centrals reach the opposite pole, okay, to the left, to the right, to the top, to the bottom, no need to mention already. Okay, so this is about Anaphase II. Okay, if we compare this one with Anaphase, the last lesson, mitosis, actually same. Okay, next would be the telophase II, the last part. Okay, now the chromosomes reach the opposite pole. Okay, now it's the turn for the chromosome decondensed in telophase II. Just now in Telophase I, we never mentioned about the chromosomes uncoiled or the chromosomes decondensed to form chromatin. But now in Telophase II, the condensation occurs and they form chromatin to become the thread-like structure. So how about the other parts? Okay, now the nucleus reappears, nuclear membrane are reformed, because as I told you, for nucleus and the nuclear membrane, 包头包尾, which means you need to mention it in the prophase. I don't bother whether this is Prophase I or Prophase II or just Prophase. And 包尾, which means at the last part, okay, Telophase I, Telophase II, or telophase. And now spindle fibers disappear. So this is the end of telophase. So once to the end of telophase II, so this cell will carry out cytokinesis. Cytokinesis involves the separation of cytoplasm. So here is a question for you. How many daughter cells are produced right now? Okay, we can see in meiosis, there is one parent cell and it undergoes two division, meiosis I and meiosis II. For each division, one cell becomes two cells. So now the answer is there are four daughter cells are produced in meiosis. So now by comparing, okay, meiosis and mitosis, for mitosis, cell divides for only one time. But for meiosis, the cell divides for two times, which means meiosis I and meiosis II. For mitosis, one cell is divided into two cells, whereas for meiosis, one cell is divided into four daughter cells. So these are the difference. Okay, boys, before the end, because you still need to draw the diagram, it's not the end of this lesson. Okay, so thank you just for sharing my slide, because it's not come to the end. You still need to draw the diagram. Okay, so your assignment, okay, will be uploaded to the Google Classroom tomorrow, okay, 29th of April at 11 a.m. So please check your assignment, and you will be given 2 days to complete your assignment. It's very simple. Okay, so you just spend this whole day, okay, Tuesday, to do a brief revision regarding to our topic today, which is meiosis. Okay, so now get ready with your logbook and your color pencils. Okay, and now I'm going to teach you on how to draw meiosis II. Okay, ready? So in meiosis I, in meiosis I, the parent cell is divided into two cells, two daughter cells in meiosis I. Now, just now I have told you, for meiosis, in meiosis I and meiosis II, the movement of the central is in two different directions. Okay, let's say this is one parent cell. Okay, in meiosis I, let's say the centrals move to up and down, up and down. So which means the equatorial plane would be the horizontal line. It's up and down the central. Okay, so for the second division, which means in meiosis II, this cell, okay, would be divided into left and right. So which means the centrals now move to the left and to the right, no longer to up and down. So remember, for meiosis I and meiosis II, the direction of the movement of centrals is in opposite. For meiosis I, centrals move to up and down, and therefore in meiosis II, the centrals move to left and right. So this is how to form the cell. Okay, so now we'll proceed to meiosis II, starting from Prophase II. Okay, so in Prophase II, get ready, we have two cells, now no one cell. two cells already, because the product of meiosis I, there are two daughter cells. So now you need to draw two daughter cells up and down. Okay. So in prophase I, everything is broken. This is the nuclear membrane has been broken. Disintegrate. Nuclear membrane disintegrate. Okay. Centrals move to the opposite pole. Now the centrals move to the opposite pole. Now from in meiosis I, centrals in our diagram, the centrals move to the top to the bottom. Now it's totally different. It's to the left and to the right. Okay, they're just moving only. Just start moving. No, just start moving. Move to the opposite pole. Okay, and then centrals begins to form. Oh, sorry, the spindle fiber begins to form, just like drawing the viscous. Okay, and then in each cell there are only two chromosomes. Previously, the cell in meiosis I, okay, there are four chromosomes, but the total cells have two because it has been reduced by half. Now, in the cell in prophase II, it has two chromosomes only. So still remember, in the previous drawing, this part, the upper part, one long, one short. Right? The lower part also same. One long, one short. Okay. So for the lower part is the darkened one, is the shaded one. And don't forget, because of crossing over, the tip is not shaded because genetic material has been exchanged. Same goes to this. Okay, so for this one tip is darkened, one tip is darkened. Okay, next one, followed by Metaphase II. So for Metaphase II, the centrals that reach to the opposite pole, so left and right, left and right. And now the chromosomes, they are arranged at the metaphase plate or the equatorial plane randomly in a single row. Okay, no longer side by side, because for chromosomes arranged side by side, this one is referred to metaphase I. Okay, are you ready? So now I erase everything. So if you cannot follow my speed, it's okay, you can just keep replaying. Okay, while waiting for the next part, okay, so remember, boys, those viewers here, remember to like this video, If it's very useful to you, okay, like this video as a support to Tansen. Okay, so come on. Now, the centrals move to the opposite pole. Now, the opposite pole here is referred to the left and right. Left and right. Okay, so which means the equatorial plane actually is the vertical one. Previously was the horizontal, now becomes the vertical. Okay, later I will erase this line. And now the the chromosomes. Okay, so now the chromosomes, they are arranged at the equatorial plane and in the single row. One long, one short. Okay, this is metaphase II. Okay, I just erase this equatorial, this imaginary line. And don't forget, this is the darkened one. Darkened one. And remember, one of the leg is different. One of the leg is different. So for this lesson, after this lesson, for my lesson, when you're following the lesson right now, you follow me to draw. But you can practice once at home. Because we need to do the same thing for 21 times. It only becomes a habit. Just like every time when you make a mistake, I ask you, hey, do correction for 21 times, 21 times. So this is the reason. If you want to train yourself to draw mitosis and meiosis 21 times, for sure, you can draw it very perfectly. Okay, and today, we still have 2 weeks. This is the third phase, the last day of the third phase of our MCO. You still have 2 weeks for you to practice. Okay, so now, and don't forget this one. Okay, so this drawing is not completed yet because something is missing, which are the spindle fiber. So now the spindle fiber is attached to the centromere of each chromosome. So the spindle fiber is attached to the centromere of each chromosome. Okay, so this is about Metaphase II. Okay, so next one we'll proceed to Anaphase II. So keep practicing at home for this mitosis and meiosis until you can draw it perfectly, no mistake. Okay, so now we go to Anaphase II. So as you know, Ana here means that Anaphase. It's not the frozen character, okay? Because previous time, okay, that is our ex student, senior. When he draw mitosis or meiosis, when he draws mitosis and meiosis, he doesn't know how to draw Anaphase. So at the end, what's coming in, what's coming out, which means the face of Ana. Because in frozen, we know we have two characters, main characters, okay, Anna and Elsa, and he draws the face of Anna. This Anaphase and that Anaphase is totally different. So Anaphase here is referred to the separation. So now the homologous chromosome, no more homologous chromosome. So what are separated? The sister chromatids are separated to the opposite poles. So now get ready with your Anaphase. So now the sister chromatids are separated to the left, to the right, to the left, to the right. Previously in meiosis is to the up, to the top, to the bottom. Okay? So now ready? So for Anaphase, your cell draw a bit elongated. Okay, elongated. It's an oblong shape. Okay, so the centrals at both sides, this is an animal cell because it contains centroles. For plant cell, they do not contain centroles. Okay, so when the sister chromatids are separated, okay, the sister chromatid is pulled to the opposite pole led by the centromere, which means the centromere always comes first. Centromere always facing towards the centrals. Okay, so now initially like that, check one long, one short. Okay, one long, one short. So when they separate, so this side to one pole and this side to another pole, led by the centromere. Okay. Initially, this part, this is darkened. This slit is darkened, right? So now, when they are separated, this slit is darkened. This slit is darkened. You see it? I draw in vehicles. And then, wait, something is missing in this diagram already. Because of the shortening and the contraction of spindle fiber, it causes the separation of the sister chromatid. So spindle fiber is attached to the centromere and pull them. Okay, so now for another cell, same. One long, one short. One long, one short. Okay, so previously this one is a darkened one. Okay, now boys, since June, I believe this is what my prediction. School will reopen after my prediction. I'm not spreading news. Fake news. This is just my prediction. Okay, I predict school will reopen after Hari Raya. So we know during the Hari Raya 2 weeks that will be our school holiday. So don't think that our school holiday we don't need to do things, stay at home, you do. No. So you still need to follow the class lesson as usual, but I will give you a priority, just 1 week, 1 hour only. Okay. So now separate this part. Sister chromatids are separated. This one is the darkened one or the shaded one. Shaded one. Okay. This one also shaded one. But don't forget this tip is not darkened. Can you see this? Not darkened, Okay, and last but not least, the spindle fiber which are attached to the centromere. Okay, so this is about Anaphase II, whereby the sister chromatids of each chromosome they are separated to the opposite pole, and these opposite poles are referred to the left and the right, because in the previous part, Meiosis I, opposite pole is up and down, now become left and right. So when we draw Anaphase II, okay, we make sure because of due to the crossing over during Prophase I, the genetic information of the non-sister chromatids has been exchanged, and therefore, when we draw the chromosomes, make sure there is a part whereby the genetic information has been exchanged. Okay, so now comes to the end of meiosis II would be the Telophase II. So now let's see when the chromosomes are pulled to the opposite pole, the resulting daughter cells would be four daughter cells. Okay, so now for your task right now, you need to prepare in your logbook four daughter cells. Okay, but before cytogenesis, there is no four cells, but we need to draw the curving in part for your plasma membrane. Okay, so while waiting for the others, while waiting for me to erase, if you are okay, ready for Telophase II, so get ready to draw the outline of the cell, the plasma membrane with curving in. So this curving in actually will form the cleavage furrow during the cytokinesis. Okay, ready? So the time now is 12:37, okay, and I will end before 1. And boys, during this MCO, everyone is staying at home, so try to read some books, okay, online books, electronic e-books related to non-academic. Okay, I know academic, some books for you is important, but non-academic reference is also important for you to upgrade yourself, and at the same time, always keep hydrate yourself. Okay, let yourself to be hydrated. Drink water while waiting for me. So drink water right now. Okay. So now in Telophase, as I told you previously, just draw like a walnut shape, whereby the center part is curving in a bit. Okay, so now there are two cells formed. So in Telophase, the new chromosomes they reach the opposite pole, and the nuclear membrane and the nucleus are reformed. So we don't bother the others one, just focus on this. Okay, and then the centrals. 1, 2, 3, 4. Now four cells. One. So four cells should be four pairs of centrals. Okay, and then spindle fiber disappeared at this stage. So we don't need to draw the spindle fiber. Okay, and now the chromosomes. So now the chromosomes reach the opposite pole, and because of the sister chromatid has been separated, so which means when we draw the chromosomes, we don't need to draw two sister chromatids joined at the centromere. Okay, one long, one short. You draw one long, one short first. Okay, so for this one, sorry. Okay, because of this part, genetic information has been exchanged, so we darken the leg. So for this one, initially, this is the darkened one. Okay, and this one also the darkened one. But this part is the white color because of genetic information exchange. Okay, don't forget nucleus reforms. Nucleus reforms. Okay, this is Telophase II. So at the end, okay, after telophase II, the cell will proceed to cytogenesis, which involves the separation of cytoplasm. So for animal cell, a cleavage furrow is formed. Okay, a cleavage furrow is formed. Whereas in plant cell, okay, the vesicles will gather at the cell equator, and the vesicles will join together to form a cell plate. This is the difference of cytogenesis between an animal cell and a plant cell. Okay, so let's see the resulting daughter cells. Now, initially the cell, which is 2n, which is equal to 4. This is a diploid number with 4 chromosomes. But now in each cell, there are only 2 chromosomes, which means in meiosis, the purpose of meiosis is to reduce the number of chromosomes by half from diploid number to haploid number. And initially we start from one parent cell, and now there are 4 daughter cells are produced. So in meiosis, there are 2 divisions, the meiosis I and the meiosis II, and end up with 4 daughter cells are produced. So this is the difference between mitosis and meiosis. Okay, so now I will come to the end of the lesson. If you have questions regarding to mitosis and meiosis, please do not hesitate to type any question in the chat box, or you can ask in our group chat, okay, so I can answer your questions. So for today's lesson, I need to, okay, while waiting for you to ask questions, okay, regarding to today's lesson, so let's do a recap regarding to meiosis. So for meiosis, it involves 2 stages. The first one, okay, so please give me the answer without typing. You just mention the name to yourself will do, or you just shout it out also can. Let your parents announce learning biology, learning meiosis, how to form sperm. Okay, so let's do a recap. So for meiosis, we have 2 stages. The first one, which is the meiosis I, and the second is meiosis II. For meiosis I, it involves the separation of homologous chromosomes. So which means at the end of meiosis I, the number of chromosomes has been reduced by half, which means from 2n to n. Whereas for meiosis II, it involves the separation of sister chromatids. Okay, in each meiosis, okay, I don't care this is the meiosis I or meiosis II. So for each stage, there are 4 phases, PMAT. Okay, so for meiosis I, because it involves the separation of homologous chromosomes. So the homologous chromosomes during metaphase I, the homologous chromosome then line up side by side at the equatorial plane. But in meiosis II, because there is no more homologous chromosomes, so the chromosomes will arrange in a single row, no need to be side by side. So remember, and one more thing before the end comes to the ending of the lesson, for prophase I is very important because crossing over occurs. And this crossing over occurs between the non-sister chromatid. So remember, it's the non-sister chromatid, different links of the homologous chromosome, and they exchange genetic information. So is that clear? And last one, you still need to know what is the importance of meiosis. There are 3 importance. The first one, okay, set it up. No need to type in the chat box or shout it out. What is the first one? Is to produce gametes, and these gametes and the production of gametes, okay, is named as gametogenesis. And the second purpose of importance of meiosis is maintain, right? Maintain the number of chromosomes, okay, as diploid number in an organisms, okay. And the last one is to produce genetic variation. So the offspring would have different physical appearance, okay. And this is about meiosis, and now officially we come to the end of the lesson. So if any question you need to ask, please type in the chat box and I will answer your question, or you can ask in our group chat, which means in our group, okay, our study group for 4S1 and 4S6. For those from different classes or even from different schools, from different states, not in Perak, not in Ipoh, from Selangor, from Malacca, or even not from Malaysia. If you have questions regarding to meiosis, you can send me the message, okay, or text comment in this Facebook in this YouTube video, okay, and you can share this video in Facebook, any platform also can. So last one, please like this video before come to the end of the lesson. Okay, so I will stop here. Goodbye, class. Thank you, class. Okay, so monitor, come on, stand up. Goodbye, class, and thank you, class. See you next week. Bye bye.